Hello and welcome to the NGS Webinar Series hosted by the Next Generation Sequencing Competence Network. Along the series, you will be hearing from scientists and clinicians from various fields of research. However, they are all connected by sequencing. Who would have guessed, We bring you to journeys where researchers are telling us about their sequencing data harvesting and bioinformatics analysis and surely results and perspectives. How do they do it? What is it good for? Watch and let us know. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We'll leave the link in the footnotes, and here is QR code for your fast access. Welcome aboard. Find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
Hello, everyone. My name is Maryia Karpiyevich, and I'm a product development scientist at SPT Labtech. It is my pleasure to present a talk today on the efficient long-read library prep in the Wellcome Sanger Institute Darwin Tree of Life project. Now, I have a very long title, and I will start by explaining what the different parts of my title actually mean and how everything came together in this project. First of all, long-read library prep refers to generating libraries for long-read sequencing. Let's look at where the long-read sequencing fits in the historical progression of sequencing technologies. So the journey, of course, began with the early sequencing methods, most notably the Sanger sequencing by synthesis chain termination method that dates back to 1977. In the early days, Sanger sequencing method relied on radioactive nucleotides and on resolving DNA fragments in large slab gels. Later on, the original first-generation sequencing method underwent development to use fluorescently labeled nucleotides. Then it got automated, and eventually it evolved into capillary sequencing. The second-generation sequencing technologies came after, and they emerged around year 2000. They're also called next-generation sequencing, or NGS, and these methods really revolutionized the field by doing massively parallel short-read sequencing. This dramatically reduced the cost and increased throughput compared to the original Sanger method. A number of different NGS platforms came on the market between 2000 and 2010, including Illumina sequences, Iron Torrent, ABI Solid, Roche 454, and so on. So short-read sequencing is still very much on the market, and we have some newer entrants in the field, and Illumina is still very much present. Now, the long-read sequences and sequencing is the third generation that came after the NGS. Emerging around 2010, technologies by Pacific Biosciences, abbreviated as PacBio, and Oxford Nanopore, abbreviated as ONT, came in, and they relied on generating much longer reads, so reads in the region of tens or even hundreds of thousands of base pairs. This is much longer than the typically 50 to 400 base pairs that are produced in short-read NGS sequencing. While the short-read sequencing is very cost-effective, accurate, and high throughput, it does have its limitations. By its very nature, NGS methods sequence relatively short DNA fragments, and if you want to assemble larger genomic contigs, it requires complex algorithms for the reconstruction of longer sequences. So often you may end up with inaccurate or incomplete assemblies that have sequence gaps in them. And no amount of short-read data can really allow for the resolution of repeat-driven gaps that exceed read lengths. Short-read sequencing really struggles with structural variants and highly repetitive genome regions, the complex genome regions. Furthermore, you need to perform rounds of PCR amplification in the course of short-read sequencing library prep, which may result in artifacts. And all these limitations can really be resolved with some of the long-read sequencing approaches. So long-read sequences do generate much longer reads, so the issue with gaps is not as severe. Long-read methods also do not require any PCR amplifications, and they typically show better resolution of complex genome regions, better genome assemblies, and better detection of structural variants. Longer reads also act as more representative elements of chromosomes if we consider the whole genome, and consequently they can produce more contiguous reconstructions of a whole genome. Now, the long-read sequencing method that we utilized for this project is the PacBio single molecule real-time, or SMRT method. Now, if we consider the preparation of library for such a method, unlike the short-read NGS library preps, we actually aim to preserve long DNA fragments, so high-quality DNA molecules that are typically in the region of 15 to 20 kb. Then the adapters that have been shaped, smart bell adapters, are added to, ligated to both ends of DNA fragments. And therefore, we end up with a circular DNA template. Then the primers are annealed and DNA polymerases bind to the circularized templates. And the PacBio sequencing really operates by continuously reading through those circular templates, which enables the multiple passes through the same sequence. And that allows the accuracy to be improved as a result. So PacBio uses this circular consensus sequencing concept to generate hi-fi reads, which are highly accurate. They're generated from sub-reads via the formation of a consensus sequencing. So we end up with 99.9% accuracy. The sequencing is also happening in real time, so the sub-reads are continuously updated, improving the accuracy as a result. Now, what's going on in the actual PacBio sequencer? So circularized fragments of sample DNA that I showed earlier, they are in solution and they're flooded across the surface of a nanofluidic chip that is called SMRT cell. So this cell contains millions of small wells, cylindrical well-shaped zero-mode waveguides. So single circularized DNA molecule is immobilized in the bottom of a ZMW. So there's one molecule per this tiny well. And once the sample of DNA is situated within the ZMV, some free floating nucleotides are added, and a DNA polymerase enzyme that is attached to the circularized template begins to copy the molecule, and the reading commences. As the polymerase incorporates new nucleotides, the light is released from the fluorescently labeled nucleotides, and is picked up by the detector. So the detector can determine which nucleotide was incorporated, and that's how the sequencing commences. Now, looking at the PacBio hi-fi sequencing, smart sequencing technology, we can see that they occupy quite an interesting space. So compared to the short reads that are short but highly accurate, and some of the other long read sequencing technologies such as ONT that are less accurate but very long, hi-fi reads actually combine the advantages of short and long read technologies because they're both accurate, about 99.9, and long, 15 to 20 kb. This combination becomes particularly useful if the aim is to sequence a whole genome, and especially if this genome is a novel genome and does not have an existing reference genome. Now, sequencing novel genomes from diverse species is the focus of Darwin Tree of Life project. So that's where Darwin Tree of Life comes in. Darwin Tree of Life is a large collaboration by multiple scientific partners that is aiming to produce high-quality reference genomes for all known species of animals, plants, fungi, protists in Britain and Ireland, so the British Isles. So this project was initiated by the Wellcome Sanger Institute, and it aims to generate the genomes of about 70,000 species and make these genomes available for research, conservation, biotechnology. It is a very exciting project, and it is a huge, large-scale initiative. Genomic initiatives of such scale become very labor-intensive. And with projects of this size, automation becomes a very attractive option because it can drive throughput and efficiency and contribute to the success of the project in this way. Now, there's many benefits of using laboratory automation for complex genomics workflows, such as a PacBio library prep preparation. Now, some of the general benefits of automation are enhanced consistency, reproducibility, and record keeping. Now, lab automation, when you program the machine, you can run the same protocol multiple times, and it will do it exactly the same way. So unlike humans that often vary the way they're doing things, robots will do the same thing day in, day out. Reproducibility can be increased because the protocol tends to be very tightly controlled, and the environment is very tightly controlled. And record keeping is easier because automation by default does collect a lot of data and does it in a seamless manner, while scientists in the lab have to do the experiment and write their notes at the same time, and often the two don't really come together very well. The other aspect of it is the elimination of operator effect and reduced risk of human error. Now, with the lab automation, because the robot is performing the actual tasks of liquid handling and manipulating labware, the human operator does not really come into play. And while in the lab, you could see that everyone does things slightly differently and everyone gets slightly different results, with the lab automation, it is much easier to standardize the protocols. And while the risk of human error is not fully eliminated in any case, it is significantly reduced with automation. Now, the one that is really close to my heart is that the use of lab automation frees the scientist to perform higher value tasks. Now, the training of scientists, even nowadays, mostly consists of teaching them to do manual tasks. And we end up with highly qualified, trained scientists spending a large part of their day doing highly repetitive manual tasks. And that is really a shame because researchers could use their time on doing much more valuable things, such as analyzing data, thinking of complex experiments, doing some of the smaller scale but very complicated experiments. And the automation can really free researchers and focus on the highly repetitive stuff, while the researchers are free to do something much nicer with their time. And increased throughput, lab automation is typically set up for doing more, so increased throughput. And this can be achieved through multiple ways. So first of all, lab automation can work 24/7 while humans can't. So if you consider a process that may take about 6 hours, a scientist will probably just run one process per day, whereas lab automation could arguably do 4 times that in a 24-hour period because it can run overnight, doesn't need to take breaks. The other aspect of it is that lab automation typically is set up to work with higher density labware. So while scientists would typically work in tubes or smaller density plates, lab automation can be set up to operate in higher density plates with more wells, or use multiple plates at a time and parallelize the processes to a higher degree than a scientist would. Now, lab automation is a big umbrella term that includes a lot of different types of equipment in the lab that is set up to run partially independently or fully independently, and can combine a lot of integrated components. Now, the special piece of equipment that we used for this project is the Firefly by SPT Labtech. So Firefly is our compact all-in-one liquid handling platform. Why did we pick Firefly in particular, and how is it good for PacBio SPK3 workflow? Now, one of the key benefits of Firefly, and that's very much something that Firefly was built on, is making it accessible and easy to use. Now, lab automation is amazing and very powerful, but often people in the lab are a bit concerned about using those large, intimidating robots. Now, with Firefly, we try to make it as approachable as possible. One big aspect of it is our user interface that is quite intuitive and easy to use, and it does not require any proper prior programming knowledge or the need to write scripts. Now, some scientists really like coding and programming, but we found that most scientists don't, and most scientists want to use something that is a bit easier. So we went on and designed a user interface that is very graphical and relies on drag and drop, and makes the protocols in a very graphical way. So on the left, you can see an example of a protocol. That's actually a first part of the PacBio protocol that we used in this project. And here you can see everything is made up of pictures, everything is quite colorful, and it is really easy to learn how to program the instrument. The other accessibility aspect is that Firefly relies on cloud, so you can collaborate quite easily and share your protocols through the cloud, share it between different scientists who are working on the protocol, but also share it between different Firefly instruments. So for example, if we have multiple sites within the same research institute or the same biotech company, and you want to work on the protocols collaboratively, you may design a protocol on one machine and then via the cloud allow it to be run on a different Firefly instrument in a different geographical location. We also allow our customers to download and use the protocols that we've developed previously, so they're all available for the new users to use. And we have a very large library of labware that we defined for the instrument that, again, the users can just download like you would do in an Apple Store. The other aspect of Firefly is that it can very reliably handle viscous reagents with the use of positive displacement technology on our dispense head. Now, in the PacBio library prep kit, a lot of reagents are made into master mixes. So master mixes tend to be very viscous, very difficult to use. So something that viscous, when you try to prepare it manually or using an air displacement technology, it ends up being not very accurate because of the column of air that is inherent in the air displacement methods, but also it just sticks to everything. It sticks to tips, it sticks to vessels. Now, on the bottom, you can see a diagram of positive displacement. So the way positive displacement technology works, unlike the air displacement technology where you just have a hollow tip, for positive displacement, you have something more like a syringe. So you have a syringe body and you have a plunger inside, and the piston rod for our dispensing head would move up and down and move the piston. And when the syringe goes into contact with the liquid, it would aspirate, and the plunger would come into direct contact with the liquid. So this makes it a lot more accurate and precise, and this also allows us to handle very viscous liquids because the liquid is aspirated into a bigger syringe and then dispensed in a non-contact manner into all the wells that are required. So with dispensing, again, the metal rod would be moving the plastic plunger inside the syringe, and the dispensing would be happening in a clean and efficient manner. The other aspect is scalability that the Firefly offers. Firefly is a very flexible instrument, so you don't need to have a very large scale project to make it worthwhile, and Firefly can scale with your needs. So one protocol can be used to run different sample numbers, because we're using variables in the protocol where you can define how many samples you're running every day, and the instrument will adapt. So you can really start small and then go bigger, or if you need to scale down again, you can scale down. The other aspect is Firefly really supports reduction of plastic use, and as a company, we're really focused on reducing plastic usage. Now, we achieved that by making good use of our positive displacement technology, syringes. The syringes dispense in a non-contact manner, so they basically just fly above your plate and dispense the liquid, which means that they never touch the sample and can be reused for multiple wells within the same plate, and even for multiple plates. And this way, we're reducing plastic. The other thing we can do to reduce the plastic on Firefly is the use of higher density plates. So, for example, while the researchers will typically use tubes or 96 well plates as a maximum, Firefly can handle high density plates such as 384 well plates. So, for instance, if you have a process spread across 4 96 well plates, you can replace them with one 384 well plate. And even when you scale the volumes down, Firefly can scale down and handle the lower volumes precisely. Okay, so how did we all come together to collaborate on automating the PacBio workflow? So it was a collaboration of 3 partners, and the PacBio partner came in with their kits, and we worked based on the method that is published and fully available. So the manual method is called Preparing the Whole Genome and Metagenome Libraries using SmartBell Prep Kit 3. So SPK3 is an abbreviation of SmartBell Prep Kit 3. From SPT side, we came in with Firefly, and the Firefly instrument was installed at the Wellcome Sanger Institute with the automation team who are joining for the Q&A. The Wellcome Sanger Institute team really did a lot of heavy lifting with preparing the samples, with running the actual protocols, optimizing the protocols, and eventually the sequencing was done by the PacBio team on the Revio platform. So it was a fantastic collaboration between the 3 partners where everyone brought in their own expertise to make this complicated workflow automated and a success. Okay, so in terms of how we staged the project on automating the PacBio workflow, so it came into several stages, and it was important for us to really make sure that the process is running well and is fully tested and validated before we run the real samples to ensure that we've done everything correctly. So first of all, the setup consisted of mapping the workflow, so essentially looking at how we would convert a manual workflow into an automated process, designing the protocols for Firefly based on the manual protocol, and preparing of the input material. This is actually a lot of effort to kind of prepare good quality DNA for long-read sequencing, and the Sanger team did an amazing job with this. Validation was the next step. So after the protocols were done, we first tend to dry test them, essentially check that the instrument would run correctly and all the timings look right. Next, we've done some water testing where you just test the protocols using water or representative liquids before you go on with the real reagents. Now, the kits tend to be quite expensive, so people like to test with water first, just to make sure that we didn't miss anything. Next, we've done a validation run where we compared the manual and the automated prep, essentially prepared some of the libraries manually and some in an automated way. And that was in a smaller scale test as a proof of principle. Another important validation check we've done is a so-called checkerboard testing. Now in this test, we checked for any intraplate contamination, cross-contamination introduced by the robot. So essentially we want to make sure that the instrument is not cross-contaminating samples in different wells on the same plate. And that is what the checkerboard test was about. Now, the third stage was the live run. So that was the real test with all the real reagents and with a test panel of 8 different species that came from the Tree of Life project. These were done on Firefly and manually, and you will see the comparison of how well Firefly performed compared to very experienced human operators. The last stage is the QC and sequencing of the libraries that were generated during the live run. So sequencing was performed by the PacBio team on a Revio instrument, and the data was analyzed and shared with us. Okay, so let's start with looking at how we map the PacBio workflow. Now, on the left, this is the diagram that came from the manual kit, the manual protocol that came with the kit. And it consists of a few defined stages. So the manual process is performed all manually, and it goes through DNA quality control, shearing and cleanup, repair and A-tailing, adapter ligation and cleanup, nuclease treatment, size selection and cleanup. So these are the stages of a manual library preparation. As you can see, there's a few safe stops where you can stop the process and then restart it. The safe stops are typically introduced in manual protocols because they tend to be very long and you may want to pause for a day and pick it up on the next day. Now, looking at Firefly automated protocols, we try to keep it as close to the manual protocol, the published protocol, as we could. And we also had some safe stop points where we could pause the process and pick up later. Now, some of the processes could happen on Firefly completely, but some of the processes, for some of the processes, we would take the plate off. For example, incubating the plate, we would take it off and incubate on the PCR machine. Now, here on the right, you can see the diagram that we have for the Firefly automated process, and you can see the processes in pink boxes are the ones that happen inside the Firefly, so such as cleanup processes, addition of master mixes, and then we have some processes in blue boxes that happen either manually or of the instrument, incubated on a different piece of equipment. And overall, we were very close to the manual process. We did change a few things just to make it work better with the automation. Okay, in terms of the equipment and kits that were used, so we used the SPK3 kit from PacBio to run the processes. We used Firefly for all the sprite cleanups and master mix additions within the SPK3 workflow. DNA quantification was done off Firefly, and it relied on AccuClear ultra high sensitivity dsDNA kit. It used some of the equipment such as LV Mosquito from SPT Labtech, Agilent Bravo, and the plates were read on BMG Amiga plate reader. For the qualitative DNA assessment, we used Agilent FemtoPulse, and for the incubations, we used a standard lab thermocycler. Okay, so in terms of validation, the first validation that we ran on the first stages that we ran was dry testing and was water tests. We assessed them by eye, and when we were happy with those, we proceeded with the first of our validation runs, and that was manual versus Firefly automated library preps with just 8 samples. So we started small, and we did just 8 samples, 8 libraries, both manually and on Firefly instrument. Now, the DNA material that we used was very homogeneous, and it was DNA extracted from the legs of a brown cricket. Now, for validation runs like this, you really want to use something well-known and a good quality DNA source that is also very abundant, and the legs of the brown cricket gave us exactly that. So when the libraries were prepared, when we ran through the whole workflow, we assessed the DNA, the resulting libraries in 2 different ways. So we looked at the qualitative assessment using FemtoPulse, and on the top right, you can see the traces here. So all these traces, you can see that they're all overlapping, and they combine all the 8 libraries from Firefly and from the manual process, and they're sitting really all on top of each other, so we could see that the libraries have the same shape and the same size. And then looking at the library yield, the qualitative assessment, at the bottom, you can see the bar chart that is showing the yields for Firefly and from the manual process. And you can see that the yields are pretty much the same with very similar error bars that correspond to the coefficients of variability. So overall, we could see that the libraries prepared on Firefly are equivalent in size and yield to those that were prepared manually, and the variability between samples is comparable between manually prepared and Firefly automated libraries. So this test told us that the way we automated the library prep on Firefly is very representative of how a person would do the library prep, and we're getting the libraries that are of similar quality and similar yield. Now, the second stage of our validation is the checkerboard analysis. And if you look on the left is the diagram of what our plate layout looked like. So for checkerboard analysis, we've had half of our wells had the same homogeneous cricket DNA material that we used in the previous test. And we had 24 samples. And this was our positive control, where we would expect the libraries to be generated. And the ones in red with a minus sign is just a elution buffer that doesn't have any DNA. And as you can see, all the green DNA material containing wells are surrounded by the red wells. So the way this test runs, it would highlight if there's any cross-contamination. So if any of the material gets accidentally carried into a neighboring well, which would be a negative well, we would immediately see it in our test. Now, after we've run all these samples, this 24 positive and 24 negative ones in a checkerboard pattern on Firefly, we went on to do real sequencing on the PacBio SQL 2E platform. So we've generated an equivolume pool of all the 48 libraries, and we've sequenced them. And on the right, you can see the sequencing results, the bar and whisker diagram. So you can see that the positive controls in green, they had an average of 93,000 hi-fi reads and a CV of about 13.6%. So you can see that we've generated reads from all the positive wells, and they are all in a fairly tight group. And conversely, the negative wells did not give us the reads. So we were quite confident that there's no cross-contamination within the plates, and the instrument can handle the plates correctly, and all the liquid stays within its designated well. It also showed us that there's uniformity across the plate, so there's no effect in the wells depending on their positioning on the plate. Okay, after we've convinced ourselves through the validation run that the process is solid and it's running reliably on Firefly, we went on with the exciting live run. So for the live run, we've got samples from 8 diverse species, and these samples came from the Tree of Life project. So in the pink box in the middle, you can see the pictures for all the species that we've used. So we've used a human sample from a cell line, we've used a mouse, and these organisms are a lot more common, and everyone has sequenced them, they have very good reference genomes. But then we went on and decided to push our system and try some of the more challenging, more unusual genomes. So we have some plant genomes. We have oak as a representative of plants. We have some green algae. We also have marine yeast. We have insects, so the ladybird and the cricket, and we have a snail. So the samples from diverse species, they are different, so you do get differences, and they're not as easy as your standard model organisms. However, we went on to prepare libraries from all of them using the same process. So the same protocols that we optimized on water and dry runs, and then on cricket DNA, we just threw all the 8 species into the same process. So they were all on the same plate. And we've prepared triplicate libraries of all of the species. by hand and then on Firefly. Now, in order to make the experiment as representative as possible, the starting material was the same. It was shared between the Firefly and the manual process. So we started with the same material. And then the master mixes and reagents for the library prep kit were shared as much as possible so that we can be confident that we have a good side-by-side comparison. Now, after the libraries were completed, we've performed a round of QC before the sequencing to see how good the libraries are. And for the qualitative analysis, we again looked at the femtopulse and on the traces generated by different libraries. So here you can see the four of the species, so ladybird, algae, marine yeast, and mouse. And the traces here consist of 3 manually prepared libraries and 3 Firefly prepared libraries. And again, as you can see, all the traces are overlapping. So the shape and the size of the traces is the same between manual and the Firefly automated ones. And the remaining 4 species here, again, oak, snail, cricket, and human. Again, you can see that all the traces are overlapping and the size and the shape is the same. So this told us that libraries prepared on Firefly are analogous in size and in shape, and therefore, from the qualitative point of view, they are very similar. Now, looking at the quantitative analysis of the library yields we got on the Firefly automated and manual process, between the for all the 8 species, I have the bar chart here showing what percentage of input was recovered as libraries. So this is shown as percentage of input. And here you can see that the Firefly and manual libraries, so Firefly in cyan and in magenta, sorry, and manual libraries in blue are showing very similar, very close yields. So they are comparable to the manually prepared libraries for the same species. Therefore, again, we were reassured that the libraries that we generated on Firefly are representative of what we would generate manually for all the 8 diverse species. After that, we went on to do the sequencing of Firefly automated and manually prepared libraries for 3 of the species. So we selected 3 different species, oak, mouse, and green algae, and generated library pools from them. So the 3 library pools were then sequenced on the PacBio Revio platform, and each pool had the 3 libraries on prepared on Firefly and 3 libraries that were prepared manually. Now for the results, we got the results back, and we were very happy to see that all 3 pools gave really good yields and produced high quality sequencing data. And then looking more closely on the different species and the sequencing metrics that we got, we were happy to see that the libraries prepared on Firefly had comparable metrics to those prepared manually. So on the table on the left, you can see the pool yields were reasonably high. So we got 77.3 from green algae, we got 98.6 from mouse, and 90.9 from oak. And then looking at all individual samples, we can see very close yields for individual samples and very close read lengths. And the quality score, the Q score, was well above 30 for all 3 species. So for green algae, it was Q37, and then for mouse and oak, it was Q35 for all the samples. Now looking on average for the Firefly and manually prepared libraries, so in the bar charts in the middle, you can see that in terms of the min hi-fi read across the 3 replicates, the manual libraries and the Firefly libraries are showing very close results and very close error bars, which corresponds to coefficient of variability. So that to us suggests that the Firefly generated libraries are close in yields, and they're also close in terms of how variable the different libraries are. And the same applies to the yields. So to the bottom panel, again, we get very similar numbers and similar variability. So overall, we were very happy with the outcomes of this project, and we saw that Firefly can generate libraries that are on par with manual libraries, but they would generate these libraries in a much more efficient way and require less labor from the scientist, and it can be streamlined to work with larger scale genomics initiatives, such as the Darwin Tree of Life. So in conclusion, we've demonstrated the successful automation of PacBio SPK3 workflow on Firefly platform. We validated that Firefly automated protocols give comparable size and yield of libraries, comparable to the manual process, similar sample-to-sample variability, and we showed the absence of intraplate contamination in the automated method. We then went on to show that SPK3 libraries could be successfully prepared on Firefly from 8 different species that came from the Darwin Tree of Life project. Thus, we've proven broad sample compatibility with Firefly. And overall, we've shown that Firefly is a viable option for automating long-read library preparation and could support increased throughput, ensure consistent performance, and reduce the need for laborious and error-prone manual handling. All these features would come really useful for a large-scale genomics initiative that requires a lot of library preparation for sequencing. At the end, I would like to acknowledge the help and support from all the people who made this project a success. So first of all, the Wellcome Sanger Institute team did an amazing job with the project and put so much effort into it. So thank you to Ben Farr, Shelley Ann Kutz, and Leslie Shirley. All 3 of them are joining for Q&As. Also, thank you to James Watts, Craig Cotton, and James Uphill, who were involved in sample preparation for the project. From the PacBio, I would like to thank Gregory Young and Camille Conner, who supported us with the kits, their expertise, and enabled the sequencing. And then from SPT Labtech, I would like to thank all the team Firefly. Too many people to mention, but thank you very much, my brilliant colleagues. And with that, I would like to thank you for listening, and I'm happy to take any questions.